Hello viewers. Today let's cover this coronavirus structure. We've been hearing this coronavirus and the disease that induces, which is known as COVID-19 disease, for quite a long time. It has changed our lifestyle and brought very tough situations to many families and lost many lives to this virus. I really wanted to know what the structure looks like and what you see here, what these things really mean. They call it as spike proteins, and there's so many words that we've been hearing from media and from doctors, from people in our social media groups. So today I want to cover what we all have to know about this coronavirus from the structure. This is what I mean by structure, how the virus really looks like and what are its components inside, kind of a dissected image in the bottom. So we'll go over what every single part of this is, and also at the end, what is our immune system and how these COVID-19 tests that uh, most labs are performing, how that works, and finally, if a company thinks about making a vaccine for this coronavirus disease, what are the options they have in their hand? Let's look at and let's go into the video. So first let's look at the structure. The previous slide you had seen a three-dimensional image, but this is a pictorial representation. Someone drew the information from that uh, electron microscope image. So this is a very simplified image that I'm going to just show and talk about and spend a little bit of time on this. But before I go into all the details of what these parts mean and what are the important proteins or the RNA, viral genome, how important it is for the virus, and what do they do. So I'm going to cover all that, but before that I want to just give a very high level overview of what are viruses and how are they different from bacteria and other organisms. Let's see first from very basic, from the textbook kind of view. Yeah, I'm basically a pharmacist. Um, so I know really some science, but still there are a lot of details that I really have to dig into books, and there are so many other published research articles to understand this. So I just want to really make it simple for you. Let's go from the very basic. So all of these viruses are parasites, meaning they cannot reproduce by themselves. So they rely on other cells, meaning human cells or other animals or plants they infect. So they are very different from bacteria and fungi, because bacteria and fungi, they are self-producing. So that is how we can see fungi and bacteria often in soil, water, and other organic wastes like sewage or within other organisms. So animal and plant viruses fall into two general classes, those in which genetic material is long DNA molecules, and those in which the genetic material is RNA molecules. So these are the two different types of viruses. Among the DNA viruses are herpes, adenoviruses, and wart viruses. This coronavirus, named after a sun-like shape observed in electron microscope, like a sun-like shape. These coronaviruses use RNA molecules to encode their genes, like influenza viruses, HIV, and other common cold rhinoviruses. So this COVID virus that causes this COVID-19 disease infects mammals and birds. You may have heard this originated from a bat. So it is very closely related to the viruses causing the earlier SARS or MERS outbreaks that happened like previously. So the coronavirus particle, this is what I mean by particles, are organized with long RNA polymers tightly packed into the center of their particle and surrounded by this protective capsids, the small ball-like structures. So in coronavirus, these proteins are called nucleocapsids. The coronavirus core particle, the circular particle, is further surrounded by an outer membrane, really very circular outer membrane envelope made of lipids. Lipids are nothing but fat molecules with proteins inserted. All these orange like and red like structures are proteins which are in the membrane. So these membranes derive from the cells in which the virus was last assembled, but are modified to contain some specific viral proteins to perform their own function, which is to multiply and survive. So that's the purpose. Like any organism, like even human, any organism, they try to survive and even in difficult situations. So that's what even the virus, bacteria, everything would do. They want food, they want to grow and multiply, and that's all they want to do. So they don't know if it's infecting a human and that human is going to die. It doesn't know. All that it wants, it needs food from somewhere. So it cannot reproduce by itself. So it has to rely on another organism. In this case, it's going happening in humans. So that is why the mutation happens. If there is any environmental stress for this virus, so it tries to mutate its own gene so that it can survive. So even we are hearing about the UK mutated coronavirus species. It could be that is why the mutations, maybe it's spreading so much and we are trying to protect ourselves and taking vaccine or maybe other medicines, so it has to somehow get resistance towards it and still wants to live and survive, so find a small mutation. So mutations always happen. Even as human, if you had seen long thousands 2000 years of history, there will be some very substantial mutations that we can physically observe. For someone like, someone living in the northern pole, their eyes are like blue, and why Chinese and Koreans are different than Americans and other Asian people. So it's all based on where they live and how they eat and what are they fighting against. So to live in those environments where these people or the organisms live, they try to mutate themselves so that they can comfortably live in that environment. So mutations have always been there. Um, there are other observations which are still need to be proved. Um, when when a virus or bacteria mutate itself, it wants to live longer. So it doesn't want to kill that host organism like in human. It doesn't want to kill the human because with that human, all these viruses also will die. So when these mutations happen, it could be towards a good side. Maybe coronavirus is trying to reduce its lethality and try to be more transmissive, meaning it wants to spread more, but it doesn't want to kill the host. So that can be, these mutations could be very transmissible, as Prime Minister of UK said, Boris Johnson said, it could be 70% more transmissible, but then they may not be as lethal as we have seen the coronavirus before. So yeah, to stop those mutations, we all have to protect ourselves the way that our governments are asking. Put mask and maintain social distancing so that we can reduce the spread and and that way we can also reduce the mutations that it's undergoing. So that's about that. So I really gone sidetracked. So just come back and look at the structure of this coronavirus, how it looks. So there are a key set of proteins in the outer membrane project. This outer membrane that project out from the particle. There are known as spike proteins. They are known as spike proteins. There is these proteins which are recognized by receptors of human, which will be infected through that. Spike proteins are the ones that come in first contact with the human cells. So these coronavirus particles are rapidly inactivated, killed by exposure to 70% ethanol or 90% isopropanol, which is a rubbing alcohol we use to wipe out boards, and hydrogen peroxide solutions, hypochlorite bleach, soaps, and detergents, as well as by UV light and high temperatures of cooking. So coronaviruses primarily infect human lung cells through a receptor. From here onwards, I'll just start showing you different graphics, thanks to biophysics website. So they have given this nice information, the schematic, how the coronavirus infects the human cell. This complete rectangular bottom is human. Imagine that as a human cell. How coronavirus is going to go into the cell and how it comes out. So all the complete life cycle explained in this schematic. Just going to go through quickly. So coronavirus primarily infect human lung cells through a receptor called ACE2. That is nothing but angiotensin converting enzyme 2. So it does some conversion, this enzyme. So that's the receptor. So that's what this uses. There's a spike-like protein comes and right snugly sits in there, and then this whole thing falls onto the membrane, and the membrane goes through the membrane. When it comes through the membrane, the outer part of this virus is gone, so the inside RNA is inserted into the inside of the human cell. So that's the first step that leads to viral infection. So the virus is then incorporated into, imagine this is a lung cell, incorporated into lung cells, and viral RNA is released into the cell. So the viral RNA molecules recruit the cellular apparatus of human, because we have a lot of cellular machinery that the virus does not have. So this virus takes advantage of human machineries, like this kind of another enzymes that helps replication of our own genome. So now the virus uses that machinery to transcript its own genome into into a viral RNA, and then it also uses our ribosome, which is a translational machinery, to produce all the viral proteins it wants. So all S, M, and E proteins are are produced for the virus using human machines, tools. So now that all these proteins are translated, and it comes, and at the same time the viral RNA also comes in the other side, so they all form a mature virion one more time, just like this, with an outer circle of the membrane. So this mature virion that try to go out of the membrane, that's called exocytosis, because it comes out of the cytoplasm, and this comes as a mature, very active virus once again to infect much millions of more cells. Here we are just showing one cell coming and going as a 3 cells, but indeed it's millions of things happening at the same time. If our human immune system is not so strong enough to fight this, it's also based on how much of the viral load that comes into your body, and how much of immune system is in your body working against it. So it's going to be a very lethal process. So that's about how it infects human and how it multiplies and grow. So next thing I want to also talk about how the basic tests are done So we've been talking about how COVID-19 tests are performed. So we just want, there are many different tests, at least two that are very, very important. And this one I'm going to talk today is PCR test, how it is done. So there is another test that is an antibody test that tests are usually done after, if you think you are infected, and after like about a couple of weeks or 3 weeks after your infection, you can go and test if your body has responded to it and produced antibodies against those viruses. That means you have immunity. But then before, even if you want to know if you are infected, so the PCR test is the one that tests uh, the um, the viral RNA, if that is present in your body or not. So what's the basic principle of this COVID-19 test that we always have been talking about? So the first procedure is someone is taking um, a swab through your nose, get samples, and second next step is to take it to the lab and they lyse the virus. They use some reagents to lyse the outer circle of the virus. You can see that that's broken, and then the RNA is out, and that will get purified, meaning it's not only one virus that is in this vial. There's so many millions of there, and all the unnecessary protein outer circles are broken and collect all the viral RNA and purify them. So the next steps are the key, that from where we talk about RT-PCR. RT meaning it's reverse transcription, and PCR meaning it's polymerase chain reaction. So combine these two steps, it's called RT-PCR. Q means quantitative, it's like still the same. So all these reverse transcription based tests, also like collectively called NAAT, that's like nucleic acid amplification test. That is nothing but you are amplifying the obtained viral RNAs. You are amplifying to predict if whether you have the viral RNA in your body or not. So that is a very common name for that. So in the RT-PCR steps, in the reverse transcription, you collected the RNA and you use a reverse transcriptase enzyme to convert that RNA into DNA. We call it cDNA because it's a complementary DNA to your RNA, and using some particular enzyme, that is reverse transcriptase enzyme. So in the last step, you amplify what you got, and then finally you're going to detect whether you have that DNA or not. So this is because you normally humans, we don't have that particular RNA that comes from virus, so it's very easy to match with the whole gene that humans have. And if this is a new gene that came from this experiment, then you're going to say, oh, you have a new uh, RNA that comes from coronavirus. So that does becomes positive. So that's the third step from all these sample steps. Okay, let's look at next thing. What I want to say. I just want to give you a very quick introduction about human immune system, and what are the parts of the human system that fights against infection. And finally, if a company wants to produce a vaccine against this coronavirus, what are the options they have? So with that, we will end this talk. So very broad overview of human immune system. So our immunity is majorly classified into two. So innate immunity and adaptive immunity. So innate immunity means you have it already in your body. That's also called first layer of defense and very non-specific. Meaning for example, your skin, when you have a rash, even if an ant or wasp bites you, you're going to produce some immune response, that is to show some redness and some itchy feeling. So that response is very, very non-specific. It could be for many different, even a small ant, or to even a big other thing that you rubbed into and paint or whatever. So it's like very non-specific. That's the first layer of defense. That's called innate immunity. So the second thing is adaptive immunity, that we also call second layer of defense, and it's very specific with a memory. I'll explain what that is. So this adaptive immunity comes after you get an infection from a virus or bacteria, and the specific antigen antibody reaction happens. For that particular antigen that comes through a bacteria or virus, your body responds very specifically to it. And so it remembers when the next time the infection comes, it already has the material or the tool to fight the same infection. So you may also take examples of chicken pox. So you may have heard that vaccine long time ago. So now that your body still has antibody, you're mostly resistant to it. So that means your antibody that has been produced when you took that vaccine, it's still fighting for you. So it knows and has the memory. So that's what it means. So also see this in our body. There are so many places that you have many of these white cells, and majorly it is in the bone marrow. So that's where most of your white cells are produced. And these white cells, we also call them watchmen for invaders, meaning they're always in guard, watching for foreign bodies like infections. That's why it's a very perfect name for them. So the one type of white cell is called B cells. So these B cells are very specific to a particular pathogen and secrete antibodies that detect the pathogen. So antibodies are proteins that bind to a foreign antigen, inactivate it, or target it for destruction by other white cells. When a person is infected by a foreign substance, B cells will begin making and excreting antibodies into the bloodstream. For viruses, it's often the spike protein that is recognized. Some antibodies that bind to the viral spike proteins can prevent the viral particles from infecting the cells. Other white cells like macrophages can engulf these compromised particles, remove them from circulation. B cells can also keep a memory, as I was telling before, and so that the response will be quicker and more efficient. There's another type of cell, white cells, called killer cells. Some of these killer cells can recognize a cell that is infected by the virus and kill those cells, meaning these cells are human cells. These human cells were already infected by virus, so that can act as a source for further infection. So these killer cells will even kill our own human cells which are infected. So for example, the phlegm that you cough up when you have a respiratory infection is full of debris from infected cells lysed by these killer white cells. So this is like a very strong defense mechanism of our body. So one of the best ways to protect ourselves against infection is to stimulate an immune system with a vaccine. So that's where the vaccine development I want to touch a little bit upon. So for example, the polio vaccine consists of inactivated viral particles. These are unable to initiate an infection but are recognized by the white cells of the immune system. Over a period of weeks, the white cells that recognize the virus reproduce in the body. These white cells synthesize and secrete antibodies that can bind to the virus in the vaccine. If the individual is then exposed to infectious poliovirus, the circulating antibodies that are already present in your body will be able to inactivate the infecting new particles. So this immunity may last for decades, as even I was mentioning the chickenpox vaccine immunity, though that differs between different antigens. So it's not the same case. It could be even within 3-4 months you may need another dose of vaccine. It depends. It depends. So when it comes to developing vaccine, it requires growing large amount of virus, often in animals or in tissue culture at large scale. So the viruses are inactivated by radiation. Sometimes these live attenuated, meaning they are still live, but then they are weakened for their lethality or function. Or sometimes the whole virus is inactivated, meaning their particles are still there, but they are still very inactivated, so that it will not produce as a bad effects as complete live uh, virus. So the viruses are can be inactivated. How they get inactivated? It can be by radiation, heat, or chemicals, or even derived from genetically weakened strains. So this another alternative is to purify not the complete virus but the isolated viral proteins like spike protein. This is safer and easier to scale up, but the immune system response to this is not as robust as is to the organized lattice of the inactive complete full virus. So these are possible, but then the vaccine produced by using these is not as effective as these. So a more recent strategy involves injecting individuals with RNA or DNA encoding for viral proteins. So this DNA is not for coronavirus, but it's a general statement that either RNA or if it's a DNA virus, we can infect or inject only the DNAs of the viral proteins that code the viral proteins. So these nucleic acids can be administered alone through a man-made vectors, or we can also take advantage of other materials that can go into the body very quickly. So in any strategy, any kind of vaccine strategy, however, enough material is needed to inject reasonable doses into millions of people. So the production is really difficult. There are so many steps that has to go through this, as we have seen in coronavirus vaccine. Since it was produced in a much faster manner, usually takes like about 3 to 4 years of work, but this time it was 8 months they could get this, and FDA approved for emergency use of it. So we most healthcare workers and people are in high risk started getting these vaccines, and there are so many other companies also in line in later stage of vaccine development process. So there will be many more coming, and they are within reach. So I did not cover the antibody test, so I'll cover sometime in the later video. Thank you so much for watching. So be all safe, wear masks, and keep social distancing from others, and have all a Merry Christmas.